Hello everyone. This is tutorial 1 for Biology and Bioengineering Summer Camp 2021. The tutorial aims at getting you acquainted with molecular biology and bioinformatics. In the first part of tutorial, we will introduce you to the basics of molecular biology by walking you through PCR, RT-PCR, and cloning. I am Akanksha, a project fellow at Proteomics Lab, IIT Bombay. I will introduce you to the basics of PCR.
The polymerase chain reaction, or PCR, has become an indispensable part of molecular biology. It has been used widely across the globe. First developed in 1983 by Carey Mullis, it allows DNA fragments to be amplified to 1 billion-fold within just a few hours. To carry out a PCR reaction, researchers begin with a solution that includes the target DNA, the DNA polymerase, all 4 deoxyribonucleoside triphosphates, the primers, magnesium ions and other salts that are required for the reaction to proceed. The target DNA can be anything from small fragments to a whole genome of an organism. In the next few minutes, we'll discuss in details the other 4 ingredients of a PCR reaction: the DNA polymerase, the dNTPs, primers, the cations, and the buffers. The dNTPs basically are deoxyribonucleoside triphosphates, which acts as a substrate for the PCR reaction. The primers are short fragments of DNA, typically 17 to 25 nucleotide long. They are complementary to the sequence on the template, that is your gene of interest or target DNA. For every PCR reaction, there has to be a unique set of primers. Therefore, primer designing becomes most crucial parameter for a successful PCR run. Critical variables to be considered while designing primers are the primer length, melting temperature, specificity, complementarity to the within the primer sequences, the GC content, etc. There are several online tools which are available which enables the researcher to design their own primers while considering the variables mentioned above, few of which are named below. Buffers and cations. The buffer solution provides suitable environment, which is crucial in maintaining the stability of the polymerase enzyme, while the bivalent cations, such as magnesium chloride, are cofactors for the polymerase enzyme. The polymerase enzyme is the most crucial ingredient of the PCR reaction mixture. Replication catalyzed by DNA polymerase is the basis of PCR reaction. Initially, E. coli DNA polymerase, clean-out fragment, or T4 DNA polymerase were used. However, during the initial denaturation step of PCR, these polymerases were degraded. This led to the discovery of heat-stable polymerase enzyme extracted from Thermus aquaticus, called as Taq polymerase. Besides Taq polymerase, which has become the standard reagent for the PCR reaction, there are several other polymerases as well. Example, Pyrococcus furiosus, PFU polymerase, or Thermococcus litoralis, TLI, or Wnt polymerase. In general, while selecting DNA polymerase for a PCR reaction, the following should be considered: the processivity, the fidelity, and the persistence of the DNA polymerase. Now, let's move forward to understanding the 3 steps of the PCR reaction. The first step of the PCR reaction is denaturation. In the first step, a starting solution of DNA is heated to between 90 degrees and 100 degrees Celsius for a minute or 2 to break the hydrogen bonds between the strands, thus produce single-stranded templates. The step 2 is called the annealing step, wherein the DNA solution is cooled quickly between 30 degrees and 65 degrees Celsius and held at this temperature for a minute or less. During the short intervals, the primers will attach to the template strand. In the step 3, the last step is called the extension step, wherein the solution is heated for a minute or less to between 60 degrees and 70 degrees Celsius. At this temperature, the DNA polymerase can synthesize new strands. At the end of the cycle, 2 new double-stranded DNA molecules are produced for each original molecule of target DNA. In a PCR run, 25 to 35 such cycles are usually performed.
Next, let's take a look at the thermal cycler, a machine which will lower and raise temperature of the block in discrete and pre-programmed steps. For any experimental design, you will have to specify the temperatures for each step of PCR cycle. These temperatures will depend upon the gene target, the enzyme, that is the polymerase enzyme, and the primers that you are using for the run. After specifying the temperature for each step, next you name the run. The next step is designating the volume that will be added to the tube. Then place your tubes with the reaction mixture in it and shut the PCR and start the run. Once you have started the run, the display will show you the step which has been running in a real-time manner.